So good evening, everyone. So the topic of today's talk is DNA fingerprinting from crime scene to courtroom. I think this topic is very fascinating and very useful in the modern science. So DNA fingerprinting, I think, heard by everyone. This is a very modern tool, and most of us knows this technique, especially through the TV show like the CID. So, where the forensic science is shown in an interesting and dramatic way, but in the real life, DNA fingerprinting is very powerful and accurate scientific method, and that had revolutionized the field of forensic investigation and also the criminal justice system. So the learning objective of today's session is we learn about the DNA fingerprinting and what are the historical background behind this technique and why this technique is known as DNA fingerprinting, what are the basic principles of the DNA fingerprinting, and it's the methodology part, like how we collect the sample and how we do the DNA analysis. Apart from that, we'll also discuss about the forensic application like crime scene investigation, paternity testing, disaster victim identification, and many more application of the forensic. And apart from forensic, I will also discuss the clinical application of the DNA fingerprinting. And each technique has some advantage and limitation. Obviously, we'll discuss the advantage and disadvantage part of this technique also. So before coming to the main topic of DNA fingerprinting, I would like to recap the human genome. So everyone knowing that the human DNA is 3 billion base pair and human has 23 pair of chromosomes. So 3 billion base pair of DNA are not, if you not packaged properly, that not fit into the small nucleus. So these DNA are packaged in such a way in the form of chromosome, and this is the condensed form of chromatin. And if we stretch the chromosome, we get the chromatin. If you stretch more and more, we get the DNA. So let's start the introduction part of DNA fingerprinting. So DNA fingerprinting is also known as DNA genotyping. And sometimes it is also called DNA profiling because we get the DNA profile after the analysis. So that's why it is known as DNA profiling. And this is why this technique is called DNA fingerprinting. So this technique is called DNA fingerprinting. Like that, fingerprints are very unique. Everyone is knowing that if fingerprinting we are using in the everywhere in as a document or in case of biometric attendance system because none of the fingerprint are the same. So like that, a DNA fingerprint are very unique, but here we are not looking the fingerprint that are in our fingertips, but we are looking the different types of unique pattern that are present in DNA and that are very important, and none of the DNA fingerprint are the same. So we can say that DNA fingerprinting is method which is used to identify the individual by looking the sample of DNA, and we are looking the unique pattern. And what are the unique pattern that I will discuss in the later slide? And I already told you that every cells of our body that contains DNA, every cells, and 99% of DNA between two human are the same. So where is the variation? So variation is only 0.1%, and due to 0.1% variation, we are different from one another. So if you calculate the 0.1% of the total base pair, that is 3 billion, so that leads to 3 million base pair. So there are 3 million base pair difference between the two people, and that difference is compared and that is used to distinguish you from someone else. So let's talk about some historical background. The DNA fingerprinting actually discovered by Sir Alex Jeffrey in 1984 when he was working in the Department of Genetics at the University of Leicester. And they found that something murky barcode like structure that are present in the DNA. And they thought that this barcode are very different from person to person, species to species. And they thought that that could be used as your identification tool, and later on they turned as the DNA fingerprinting. And the first case that was solved by this technique was the paternity disputed case, and later on this technique was used in the various area of the forensic science. It is not limited only the forensic science, but also in the various field of the clinical research also. So there are two great personality in the DNA fingerprinting. One is Alex Jeffrey. I already talked about him. And other one is the Dr. Lalji. So Dr. Lalji is known as father of DNA fingerprinting in India. And the Alex Jeffrey is the father of DNA fingerprinting. So what are the principles? What you are looking in the DNA fingerprinting? So in DNA fingerprinting, we are looking the repetitive DNA sequence, and sequence are present at the STR locus and the VH. To such variability, each individual are unique from one another. So we can say that the repetitive DNA sequence leads to the genetic variability. So let's talk about the DNA sequence. Like DNA sequence are generally two types. One is the single copy DNA sequence and other one is repetitive DNA sequence. Repetitive DNA sequence are further classified into two categories. One is a moderately repetitive DNA sequence and other is highly repetitive DNA sequence are present on the satellite DNA. But we'll talk about the moderately repetitive DNA sequence, among which the microsatellite and the mini satellite, these two repetitive DNA sequence are used in the DNA fingerprinting, and that is called tandem repetitive DNA sequence. So what do you mean by tandem? Tandem means the some sequence that are present one by one in the genome. So that is present in the tandem manner. So that is called tandem sequence. But if the sequence are present and after some DNA sequence, other sequence is present, which is called interdispersed sequence. But in case of DNA fingerprinting, we are looking at sequence in the genome. So there are two types of sequence. One is the mini satellite and other one is microsatellite. Mini satellite is also known as VNTR. That is a variable number of tandem repeat, where the repeat size is up to 100 base pair. Like here, the 100 base pair are repeated 4 times in the genome. But in case of microsatellite, it is called short tandem repeat. Here the repeat size is only 2 to 6 base pair. And they are repeated several times at the particular locus. Like at this locus, these repeat unit STR are repeated 8 times, but at the particular locus, at different locus in the genome, they are repeated 20 times. And the number of repeat is vary from the one person to another. So let's like here is 3 people, and I would like to see the repeat at 2 different sites. So at same site, we are looking the same tetranucleotide repeat, that is STR repeat of 7 times in that person, but at the same site, in other person, it is repeated 4 times. In other, it is 12 times. So the number of repeat is not same in any of the individual at any of the site. So that are the basis of DNA fingerprinting. So what are the technique that is used for DNA fingerprinting? So the technique that depends on the type of your objective and the type of the sample. But the DNA fingerprinting are of 3 types. First one is the RFLP. This is a very traditional method and is also known as restriction fragment length polymorphism. And it is very time taking, it is a tedious method. We need the lots of DNA to run on the agar gel. So this method is not currently used by any of the lab. And the last one is NGS. This has a high discriminating power and they have enhanced accuracy. But I will talk about the STR and SNP. This is the gold standard method that is used in every lab for the DNA fingerprinting. It is the PCR based method, and this method come in existence after the discovery of the PCR. Advantage of this method is that we can amplify the DNA even the small amount of sample. We can actually, in case of forensic science, sometimes we have compromised sample. So if we do that RFLP, we could not get the sufficient of DNA to do the RFLP. So PCR method is very good method, and we can get the DNA profile from the less amount of DNA and the less time taking. So this is the gold standard method, and this method is used in every lab. Types of sequence variation in the DNA. One is the sequence polymorphism. Sequence polymorphism means there is only changes in the single nucleotide, so that is called sequence changes, and that can be detected by the SNP. Length polymorphism. Here is the changes in the length, like the tetranucleotide, that is STR. They are repeated 3 times. There are 3 repeats, so length is more. But if repeat are 2 times, the length is less. So this is the length polymorphism. And in case of STR, we are looking the length polymorphism. But in case of SNP, we are looking the sequence polymorphism. Compare our genetic information with the just like library information. If we compare our body with library, so each cell of our body is just a book of the chapter, book of that library. And each cell contains nucleus, like each book has own chapters. And the chromosome are present inside the nucleus, and each page number are present on the chapter. And the line of the page is a locus. So we are looking the DNA profile at the particular locus or the particular chromosome. They have different type of locus, and there's some limited locus. We are looking the DNA profile on that some limited locus for identification purpose. So before starting to DNA fingerprinting, what we need? So we need the biological evidence, and this biological evidence we can collect it from the crime scene. There are various type of evidence. So what is evidence first? So evidence means anything that links the crime. In case of criminal investigation, so that is called evidence. But we have to do the DNA fingerprinting. So I will talk about only the biological evidence, not any other physical evidence or any other evidence. So biological evidence could be any biological fluid that could be collected from the crime scene. That could be the blood, semen, hair, or saliva, etc. And the second aim after the collection is the preservation. How we preserving these evidence and how we maintaining the chain of custody. What do you mean by chain of custody? Chain of custody is very, very important in the forensic science. Actually, if I do the work, so I have to hand over to other, so it should be documented in the proper way. And the last one, to avoid the contamination. Contamination is a very big issue. If the sample is contaminated, our DNA profile will be mixed and we are not able to interpret our result. And the most important thing in the forensic science is the principle of exchange. Principle of exchange says that when every person come in contact with any object or any another person, they transfer the material. And actually, this principle was given by Locard. So this is also known as Locard exchange principle, and this is very, very important in case of forensic investigation. These could be the source of biological evidence for DNA profiling. And these are the reference samples. So we have collected the reference sample, either the cheek swab like saliva, or we can collect the blood either in the EDTA vial or in the FTA card. The FTA card is very like it just like the tissue paper or filter paper, but it is prepared in such a way that your DNA, your cell is lysed and DNA is immobilized to this membrane, and you can preserve this card for a long time, even at room temperature. So this is very useful for the segment of the biological sample on the FTA card. So this is the process of reference sample collection from the suspect or for any paternity test or other test. So everything we collected from the crime scene. So what is the purpose of crime scene investigation? To collect the evidence, and we have to follow 7S during crime scene investigation. These are the 7S. First S, securing the scene. We have to secure the scene so there is no interruption. Separating the witness, scanning the scene, and then we have to seeing the scene, sketching, searching of evidence, and then secure and collecting the evidence. All has been done during the crime scene investigation. Then how we collected the sample? We'll collect the sample, but what are the way for the sample collection? So first we have to identify what I am going to collect it. So we have to report the evidence description. What is it? What are the origin of this evidence? From where we are collected? And we are maintaining the chain of custody into each and every step of forensic investigation. After collection, the storage, I already discussed earlier, we should be properly collected, transported, and stored at proper temperature before the DNA testing. So packaging is also important during evidence packaging after collection. So there are two main goals of evidence packaging. One goal to protect yourself. See, during packaging, if you are not properly packed, so we may get the contamination or we may get the infection of any virus like HIV or hepatitis, or we may get the exposure of any mold. And also our purpose to protect the evidence, not only ourselves, to protect the evidence. The evidence should not be contaminated, should not be cross transferred, or should not be deteriorated. So these are the aim for the evidence packaging. Packaged. Does your evidence is needed prepared for packaging? How we know? So in biological evidence, we ensure the samples should be air dried. Second thing, we should never use the plastic container. Always use the paper container for the biological specimen. Like what can happen when the biological evidence is packaged in the wet condition? If pack the biological like blood stain in the wet condition in that envelope, what I am seeing, I am seeing here the evidence is leaking through this exterior container, and due to which there is the sample deterioration, that could be sample loss or the contamination issue. So packaging of biological evidence is also very, very important. So before DNA fingerprinting, what we are doing, like if we get that, I have to extract the DNA from any biological fluid. So before that, the fluid is of what fluid? Is it blood or is it semen? So we have to identify first fluid. So we are doing the presumptive test. Presumptive test is not the accurate test, it is presumptive, but they give a somehow idea about the biological fluid, what we are doing. Like for the blood, there are various presumptive tests like color test, chemiluminescent test, fluorescent test, and the several confirmatory tests like tachyamia and Teachman test. So these are all table form of all the color test, but most commonly we are doing in the lab for the blood identification or the phenolphthalein test. This is also known as Kessel-Meyer test. It's discovered by the Kessel-Meyer. So we are doing the phenolphthalein test for the blood identification. I'm just showing you how we identify the blood. Like this one is the dry stain, and I have to identify is it blood or not. What we'll do, first we'll take a swab on the cotton bud, and then we add a drop of phenolphthalein solution, and then we add the drop of hydrogen peroxide. If it turns pink, it means blood is present. If there is no change in the color, so it means blood is absent. So this is the just presumptive test for the blood identification. And these are the chemical reaction. The phenolphthalein generally present the reduced form is colorless, but when it come in contact with blood, they give the colored pink compound because phenolphthalein is oxidized in presence of heat. Other tests, there are chemiluminescent test. This is the most sensitive as compared to color test. But this test is only, but it has also some disadvantage. We need a dark because here we use the luminol. But this test is useful when we try to attempt to clean the blood traces from any crime scene. And then we spray the luminol. Advantage of this technique. Okay, so now blood is present, but blood may be of any species. This blood may be of human, or blood may be of any species like goat or cow. Then what we did for the species identification, we are doing the tube precipitin test. This is the immunochromatographic test, in which we are adding the different antisera against the different species. And if the precipitin ring is formed against that species, then they link to that the blood is present of that species. So this is the origin of species identification. If the blood is of human, then we go for the blood grouping. If we ask which blood group are present, then we do the blood grouping test. But this test is somehow different from the other normal blood grouping that we are doing in the pathology lab, because here the blood are mostly present in the dry form. So we have to elute the blood from the dried sample. So method is called absorption elution. And if the agglutination is present in the A grouping, so this is A, if the agglutination is present in the B, if the agglutination is present in the A and B both, that is AB, and the no agglutination, that is co-blood. The semen test, this is also presumptive test, also immunochromatographic test. And similarly, we're doing the kit came in the market and we're doing the PCA test. And if PCA is present, because PCA is the antigen that are mostly present in the semen in the high quantity, and the band depict the presence of semen in the particular cloth. Now let's come to the DNA fingerprinting after the presumptive test. And the first DNA fingerprinting that was produced was the RFLP. So like if we identify the particular stain in the blood, so what we did, we are doing the DNA extraction from this blood sample. And after the DNA extraction, we are doing the restriction digestion. And then run this DNA fragment on the agarose and do the southern blotting. So transfer of gel to the nylon membrane. And then you incubated the membrane with the radio level probe, and we check this radio autograph by using exposing the X-ray. So for example, I have 4 samples. One is a sample that I collected from the crime scene and 3 suspect, and I have to match it. So similarly, we'll do all these things and we get this type of the pattern. So here I am looking that I'm able to see that the suspect 2, DNA profile of suspect 2 is matching with the crime scene. So in this way, we are able to identify the DNA profile. What is the old method? We need the lots of DNA. So nowadays, the method that we are using is the STR based method. So STR is a short tandem repeat, and this is a PCR based method, and we are using the codage database. So initially there were 13 locus. So these yellow highlighted are the 13 locus. We are looking the repeat at this locus by using the DNA amplification kit. And in 2017, 9 more locus was added. And all these locus during DNA profiling, that now kit, they have 20 or more than 20 locus, we are able to see the DNA profile. So these are the basic steps of any DNA profiling: sample collection, and then DNA extraction, quantification, amplification, and then genotyping and the data interpretation. Similarly, we extracted the DNA like VNTR, and after the DNA extraction, we amplify this DNA fragment using the kit. And after amplification, we do the electrophoresis, but that is the capillary electrophoresis that we are doing in the Sanger sequencer or the genetic analyzer, and we visualize the profile as a peak. Pattern of DNA profile, like this is the DNA profile at one locus, looks like this one. Like this is the DNA profile of mother, DNA profile of child. So inheritance pattern says that the 50% we are getting the DNA profile from the father, and we are getting the 50% from the mother side. So similarly, this is the maternal side, here repeat is the 6, here repeat is the 8. So we get the 2 peak, one is from the maternal side, this is the 6 repeat, so they designated the 6 allele, and the 8 peak, 8 repeat, so they designated the allele number 8. So common STR that used in the human identification are autosomal STR, YSTR, mini STR, and mitochondrial DNA testing. Most commonly used method are the autosomal STR for any identification purpose. But if we have to establish the patrilineal ancestry, then we do the YSTR. And if we have to establish the matrilineal ancestry, then we do the mitochondrial DNA testing because they enter from the mother to daughter. And sometimes the DNA sample are very compromised. We are not able to get the DNA profile. Then we do the mini STR because there's a small fragment of flow are present in this. So this is just the DNA profile, how it looks like. After the analysis, the DNA profile looks like this way. And these are the locus. And all the locus, like locus DS81179, then 1215, 2829. So we are looking the DNA profile of the genetic sample. Compare the electropherogram. Like this is the DNA profile of the evidence sample. And this is the DNA profile of suspect 1. Okay. So now we are able to compare the electropherogram generated from the evidence sample to the suspect one. So like at the locus 1, the DNA profile is the 1215, but at the same locus, the peak is only 40. Then we look in the other locus, 2829, but here it's 2931.2. Similarly, we look one by one locus, and none of the locus is matching to the evidence sample. So we can exclude that the suspect 1 was not responsible for the crime scene because that's not matching with the evidence sample. Similarly, evidence sample are the same. Then we try to match with the other suspect. Then we extract the DNA profile from the suspect 2 and try to match it. So at the each locus, the DNA profile of the evidence sample is same as the DNA profile of the reference sample. So we can't exclude. It means the suspect 2 is matching with the evidence sample and they are present at the crime scene. I'm just giving a case scenario of a disputed paternity test. Like Sarah and John, they were in relationship for 3 years and they lived together for 2 more years. Before separating shortly, Alex was born. Sarah claims that John Alex is biological father, but John had doubt about his paternity and has requested a DNA paternity test to confirm his biological relationship with Alex. So in this test, we are doing the same thing. We extracted the DNA profile from all the three samples like mother, father, and the child. And then we amplify the DNA. And after that, we do the genetic analysis. And result we are interpreted by the electropherogram and we make the tabular form of electropherogram to compare the result. So like this is the DNA profile electropherogram generated by the mother, that is Sarah. This is the DNA profile of father, John. And this is the DNA profile of child, Alex. So what we are seeing here, the profile of each locus, like the first locus, the DNA profile of Alex is the 10 and 14. So 10 allele definitely comes from the father John because father John has 10 and 12. And 14 that comes from the mother because mother has 14. Similarly, at the second locus, 30, 32.2. So 30 maybe come from the father, but 30.2 always comes from the mother because so 30 couldn't come from the mother because 30.2 is only in the mother. So mother child gets 30.2 from the mother and 30 from the father. So similarly, we check at all the locus and they are inherited 50% from the father side and 50% from the mother side. So we can say that Alex is the biological son of the John, and John is the biological father. It proves that. Similarly, in case of sexual assault case, like some case, like we have the exhibit garment of any victim, and I have to identify if sexual assault has taken place or not. So we extracted the DNA and after amplification or after genetic analysis, we make the electropherogram, the tabular chart. We separated the male fraction because there is a method for DNA extraction for the male and female fraction during extraction process by the differential extraction method. And we're able to get the different fraction of the male and the female and we get the DNA profile. So the male fraction is matching with the accused. So the sexual assault has taken place by this accused. So similarly, we can also use in case of the unclaimed body or unidentified body, like we have just one bone piece, and I have to identify the bone piece of who. And if someone claims that, then we do the fingerprinting from that accused, from the deceased father or deceased mother, and we try to establish the DNA identity, and that has been established by the somewhere we couldn't get the DNA profile, then we have to use the mini father kit. And the bone piece, we are able to match it with the father. So we can say that DNA fingerprinting is very, very important and very, very good tool. Now talk about the NGS. NGS is used to create the forensic DNA profile when the traditional DNA profile couldn't solve it. But this technique is used not too much. This is only used for the research purpose. It is not the routine practice. In the routine practice, we are just doing the STR DNA profiling, but if you want to know the sequence inside the STR, or if there's any mutation inside the sequence, then we do for the NGS. So these are some forensic applications of DNA fingerprinting. So DNA fingerprinting solved the several cases like Rajiv Gandhi Hatyakand case or the Tandoor murder case. In case of sexual assault, Nirbhaya gang rape case that has been solved. That will be useful in the paternity test, in court immigration test, and we are able to use this DNA fingerprinting in case of mass disaster, like any earthquake or any tsunami. So in case of DNA fingerprinting, if you use, then we are able to putting the piece together. Apart from the forensic application, DNA fingerprinting is not limited only to the forensic science, but it has various clinical applications. This technique is used in case of genetic disease diagnosis, research, transplant matching, pharmacogenomics, and the cell line authentication. DNA fingerprinting is also useful in case of the cancer diagnosis, like we are able to identify the mutation in the cancer gene. And if you identify the mutation, we are able to guide the therapy. Printing is used in cancer diagnosis also. Like in case of Lyme syndrome is type of inherited cancer in which there is the colorectal cancer, but they are inherited. And the main feature of this tumor is they have the MSI. MSI means they are microsatellite unstable tumor. Due to deficiency in the mismatch repair protein, the repair protein couldn't repair the DNA mismatch, and that tumor leads to the MSI. And if the tumor are MSI, there will be very good prognosis of immunotherapy. So these are the method of MSI testing. We are taking the tumor tissue and the normal tissue from the colon, extracted the DNA, using the PCR kit against the MSI. And this is the tumor, they have MSI high, this is MSS, this is a stable tumor. So that is repaired by the DNA repair protein, but where the mismatch occurred, they couldn't repair by the mismatch repair protein, and this tumor it leads to the MSI high. And the tumor that is MSI high have the immunotherapy recommended, a very good prognosis. It also useful in the several neurological disorder like repeat expansion disorder. So these are the some example of repeat expansion disorder like Fragile X syndrome, Huntington's disease, ataxia, and the dystrophy. And these are the list of different type of neurological disorder where the repeat length, like in case of Huntington's disease, the repeat is CAG, and the normal repeat unit is 10 to 35. But if the number of repeat is more than 35, that leads to the abnormal. Similarly, in case of other genetic disorder, the number of repeat, the normal range is given, and beyond that, that leads to the repeat expansion disorder. It is also useful in the several prenatal and neonatal applications. So DNA fingerprinting could be used to analyze the fetal DNA in the maternal blood, or we can able to identify the genetic abnormality in the fetus. It is also useful in the newborn screening. We can able to identify the genetic disorder at birth also. Apart from the prenatal testing, like you can do the DNA fingerprinting can be used in case of sickle cell anemia, where the mutation in the hemoglobin gene, and we are able to identify the mutation, and we can support the genetic counseling to that family. Also, for the cystic fibrosis, there is any mutation in the cystic fibrosis gene, like CFTR gene. In case of cystic fibrosis, there is a thick mucus formed and they block the airways. So, if you were diagnosed with the mutation in the CFTR gene, then we use, and this method is mostly used in case of in vitro fertilization before the IVF. DNA fingerprinting is also useful in case of organ transplantation. So before organ transplantation, we have to match the HLA. So that is good. And the second thing, after the transplantation, we have to monitor the graft. So this is very useful in case of organ transplantation also. DNA fingerprinting could also be used in case of if the sample are mislabeled or mishandled, they're not proper labeled. So with the help of DNA fingerprinting, we are able to identify the mislabeled sample also. And it is also useful in the cell line authentication because it is very useful. Because cell line authentication, cell line is very model for various types of human disease like cancer, or we have for the vaccine production, we have to choose the particular cell line. But if cell line is not correct, so we could not get the recommended protein for the therapeutics. So cell line authentication is very useful, and DNA fingerprinting helps to identify the cell line. And it is also useful in case of hospital, the child swapping case always happen. So DNA fingerprinting solve the child swapping case in the maternity ward. We are able to identify the twin zygosity by DNA fingerprinting, monozygotic twins or the dizygotic. Now the challenges in the future prospect. Still, each technique has some challenges in future aspects. So DNA fingerprinting also has some ethical concern like the privacy and data security issue. And the cost is this technique is still costly and it is not accessible by each and every lab. Most of the forensic lab is doing this type of testing. And future prospect, we can say that we need the global database for the criminal investigation of our unclaimed, unidentified body. So we need the database, but not now in India. And the future is the AI driven in each and every field. So we can say that DNA fingerprinting has revolutionized the field of forensic medicine and the scientific research. And it's a very scientific method, reliable method, and unbiased technique. I can say a man can lie, but science doesn't. Thank you.
Thank you, madam.